So first of all, welcome to our 2022 student competition. Thank you all for taking the time to be here with us on a hopefully nice Saturday morning. I know that that's what we have here in Ontario, and I join you from our office here in London, Ontario. We will begin promptly just because we do have a tight schedule that's pretty precise right down to the minute, just to make sure that we get all the teams in and allow the time for the judges to deliberate while we have a presentation occurring. A few housekeeping items. I encourage everyone to watch our social media and our website for any great things that we have happening. We always have something on the go, whether or not it's around support, education, research, and we encourage everyone to follow along with us and join us on the journey. A couple notable things are that we have our Hats for Hope campaign that's on right now. And on October the 24th, we join everyone in sharing a hat and a picture and using the hashtag Hats for Hope to help us spread awareness about Brain Cancer Awareness Day. So if you don't have a hat and you would like to purchase a hat, we've got a great one again this year. And if you would like to use your own hat or a previous hat, then we welcome that. Just please spread the word and let's raise some awareness about Brain Cancer Awareness Day. We also have an upcoming webinar on October the 21st, or I'm sorry, the 25th, which is a Canadian perspective, the use of tumor treating fields and management of brain tumors, that will be presented by Dr. David Roberge. Also upcoming in October, we will be launching our 2023 Research Award Program. So in October, we generally have our fellowships and studentships that are called out. So if you are eligible to or know someone who may have interest, then please do watch for that to be coming soon. Everyone is currently muted, and with the exception of the judges, we have cameras off. We will ask that each team has a presenter, and when it is your team's turn, please unmute and show your screens so that we can see who's presenting. After everyone has presented, we will have Dinah present, who is the only question and answer period that we have. We do have questions after the student presentations. However, that is open for the judges just because we have 5 minutes and they need to ask some key questions to be able to pick the winning teams at the end. So again, please ensure that you're unmuted when the judges are asking their questions, and judges too, to unmute yourselves. What else do we have here? This too will be recorded. It's already being recorded now for anyone who wishes to view it after or didn't get the opportunity to join us. A little bit more about why we're here today. We had 13 teams that submitted through letters of intent and interest to participate in today's event. We would have loved to have everyone, however, it made for a pretty long day of presentations. So the judges took the opportunity to select the top 6 teams who would move forward to present for us today. Today, 6 of those will move forward, and we thank everyone who took the opportunity. I know that there's some teams that submitted through letters of intent that join us today as participants and are viewing, and we thank you all for taking the time to submit through your letters of intent. I'd like to take this opportunity also to thank many that have made this day possible. This has been going on for several years, and Dr. Ranger, Dr. Motsters, and Dr. Megacy have always been great supports to me throughout this journey in terms of judging and providing cases and feedback as we move forward. So thank you all so much for taking your time out of your incredibly busy schedules to be with us on a Saturday morning to move this competition forward. I'd also like to thank, again, everyone who submitted through letters of intent. Special thanks to Dr. Joe Megacy, who has provided us the case today for the students to review and to present their topics on. So thank you so much, Joe, for taking the time to provide us a case. Special thank you to our founders, Dr. Del Maestro and Pam Del Maestro and family, for making the awards at the end of this, the monetary awards for the winning teams possible. So thank you so much. And I'm not sure if they're on yet, but I know that they did have plans to attend. Monetary prizes will be given to the top 3 teams. So we have $1,000 for the first winning team, $750 for the second team, and $500 for the third. It's very strange process that happens, but in terms of keeping everyone on track, we do have a pretty tight timeline. So each team will be provided 8 minutes to present. And we will, sorry, I'm just trying to set a timer here so you can hear what it will sound like. So after the 8 minutes, a timer will go off. Teams have been shared to make sure that they're watching their time so that they know when the time is coming close. Once the timer does go off, we encourage everyone to wrap up their current thoughts immediately so that the judges can then begin a 5-minute timer as well. The judges too will be timed, and we ask the same thing of the judges, that they conclude their question as quick as possible once that timer has gone off. So just so that people can hear and they know what will sound like when the timer goes off, it will sound much like this. So yes, that will happen so that everyone can hear. Was everyone able to somewhat hear that?
I didn't hear that, Sue.
Okay. I will make sure that let's see if we can. Still nothing?
No, sounds like silence.
Okay, so what I will do then is I will have my camera off for the presentation. When my camera comes on, that will mean you have 1 minute left. And we will we'll do it that way, I guess. In the meantime, if I can find a better way to make the audio a little bit louder, then we'll do that. So yes. In the top right-hand corner of your screen, you'll notice that it says View. If you wish to select just the speaker rather than the gallery, if you haven't already, then this will show your screen with just the individual who's speaking rather than all of those who are involved today. Presenters, please make sure that if you are the one presenting, that your name is actually coming up on the screen as your name, so that I know who to make the presenter. Without further ado, Dr. Megacy, can I pass the baton over to you to explain a little bit more about the competition and the case itself?
Sure, I can do that. Can you hear me, Sue?
Yes.
Okay, so I'm just going to be verbal, since my internet, if I put the camera on, it sometimes destabilizes. So I'll just be man of mystery here, but I will describe the case. The spirit of this competition is a case-based, in the sense that we give the students a case, and then a little bit of management, but leave the ultimate questions and themes to the students to come up with some ideas, and we give some suggestions, but ultimately the students pick their own avenue of inquiry. So this year we picked a bit of a different case. Usually we present cases that have frankly malignant brain tumors. But this year we decided to pick a type of tumor called a meningioma, which generally are benign, but not always. They usually present with a longer history, although in this case it was kind of an intermediate length of history. The patient was 55-year-old software engineer, and he had forgetfulness for about 6 months. But more recently, he had noticed some new problems, balance problems, and some headache. When he was examined, his mini mental examination was impaired, and he had some objective ataxia. So as part of the workup, he underwent a CT scan of the head and magnetic resonance imaging of the head, which showed a lesion in what we call the atrium of the left ventricle. Ventricles, as people probably know, are where the cerebrospinal fluid is made. In this case, the patient had a lesion in the ventricular system. Interestingly, the neuroradiologist who first reviewed the imaging was actually favoring this tumor to be not a meningioma, an intraxial tumor, actually a glioma, high-grade glioma. But subsequently, he underwent an operation which showed that, with a resection of the lesion, which showed that it was a meningioma. The pathologist, though, when they analyzed the tumor, graded it as WHO grade 2, which is not really a benign tumor, but not frankly malignant either. It's a tumor with some interesting features, and it's generally called atypical. After the surgery, the patient met with the neuro-oncology team, including the neurosurgeon, neuro-oncologist, and radiation oncologist, to discuss the next steps in the treatment. So the questions that we suggested that the students look into were biology of atypical meningioma, response factors that might be evident in the biology, something called methylation status, which is a newer concept around meningiomas, other treatment modalities that include radiation therapy or even experimental therapies that students might come up with. And then finally, we also suggested quality of life topic as well. So I think, Sue, we got a real interesting mix of proposals, and I'll leave it there for you to start the process. Thank you.
Thank you very much, Joe. I appreciate that. I'm just going to try one other thing to see whether or not you guys can hear this timer. And if not, we'll go back to the original plan.
My cat can hear it, Sue, but it's quite high frequency.
Okay, so let's just do it that when you see my face come onto your screen to all the teams, that is your 1 minute warning, and start to kind of do a bit of your wrap up. Same with the judges as well. That way that if people can or can't hear it or don't have their cat around, then we can still be able to notify everyone. So yes, so we will get started. We have teams today who are presenting from Queen's University, McMaster University, Western University, McGill University, University of Montreal, and Memorial University of Newfoundland and Labrador. So welcome to all the teams. Thank you so much. All of the teams, once they come onto the screen and take the reins, they're asked to introduce themselves, their team, and then roll into their presentation. So without further ado, we will pass the mic over to Queens, who will be presenting first. And we have Dhruv. So Dhruv, you can start to share your screen. And I can see it there.
Okay, perfect. All right, I guess we can get started. So hi there, my name is Gurdat, and this is my colleague Dhruv. We'd like to thank the Brain Tumor Foundation of Canada for letting us showcase our presentation for this case study today. So we'll be discussing developing a radiogenomic classification of atypical meningioma using DNA methylation status and machine learning. So to give you a little background, meningioma is the most common brain tumor in the world with a survival rate of over 67%. The recurrence probability is 10% after 5 years, and meningioma has 15 subtypes and is also classified into 3 different grades, including benign, atypical, and anaplastic.
Looking into the clinical workflow of meningioma, as we learned with Andrew's case, meningioma patients typically begin with symptom presentation. This can include nausea, cognitive deficiencies, and movement issues. And then this typically progresses towards a clinical evaluation, where radiological scans are taken to try and diagnose the issue. MRIs are typically the focus of diagnosis, and this is what Andrew underwent. Following this, radiologists evaluate the scans to diagnose a tumor and manually segment the tumor location. One of the key challenges with this, however, which we aim to address in our proposal, is the heavy time consumption involved with this manual diagnosis, which can delay treatment by days or even weeks. Moving along in the workflow, Andrew underwent a biopsy or resection procedure to remove the tumor mass, which can be invasive and costly when performed unnecessarily, another challenge we aim to address. Next, the resected tumor sample was sent to a pathology lab for tumor grading into the 3 subtypes, which brings us to our final challenge we aim to address. The current grading protocol by the WHO is not standardized, and it relies heavily on subjective evaluations of radiologists while lacking critical information on tumor characteristics, specifically from a genomic lens. And then finally, the tumor is treated and monitored for recurrence through clinical follow-ups. So keeping these 3 challenges in mind, our research proposal aimed to utilize machine learning to develop a novel and robust protocol for grading meningioma tumors. We hypothesize that the use of machine learning will allow us not only to predict the methylation status of genes from radiomic features, which we'll touch on shortly, but also accurately classify the 3 types of meningioma separately using a radiogenomic approach, providing an individualized and standardized classification. A quick overview on machine learning. We've probably all heard of it. It essentially refers to algorithms that can identify trends in large datasets using important features, understand the patterns in these trends, and then apply them to new but similar data. As we have seen over the past decade in research, ML has actually revolutionized the field of biomedical sciences, with many applications in cancer research. For our purposes, we chose to focus on not only applying ML for radiological, but also genomic analysis of tumors. One of the key benefits of using machine learning is that it not only decreases the time required for radiologic analysis, but it's also likely more accurate and consistent compared to radiologists.
Yeah, so why use methylation? So epigenetic alterations like DNA methylation have been identified as a contributor to the development of cancer. The mechanisms of alteration in DNA methylation includes hypomethylation and hypermethylation, both of which are well cited and recognized in the literature for the onset of meningioma. So how does methylation actually impact meningioma? So an existing study demonstrated that among the 12 cancer-related genes they analyzed, the results of the study do attribute DNA methylation playing a crucial role in the development of meningioma. In grades 1 and 2, about 3 genes were the most methylated, and one particular gene had a significant correlation with the WHO meningioma grade and could be considered as a possible indicator of tumor aggressiveness. In terms of standardization, the problem with the WHO gradings for meningioma is the lack of biomarkers. Histological images and MRIs are not enough for an accurate diagnosis. In fact, some meningiomas, while maintaining a benign histological profile, can display malignant behavior. The incorporation of biomarkers in the grading process can help to stratify patients as there's significant clinical and genetic overlap among the different meningioma grades. So the way DNA methylation can be used in conjunction with imaging is radiogenomics. Radiogenomics combines radiomic data and genetic data to provide a precise diagnosis and assessment of the prognosis. The utilization of radiogenomics is time efficient, as establishing the region of interest is quicker, it's much less invasive as biopsies wouldn't be needed, and it's cost effective when compared to conventional genome sequencing. Additionally, the information of the whole tumor would be available as opposed to just a biopsy, allowing for greater understanding of tumor heterogeneity. So our specific aims include segmentation, where we develop a machine learning model to segment tumors from type 1 weighted, type 2 weighted, and FLAIR MRIs of various patients and extract the corresponding radiomic features. In terms of methylation, we want to determine the distinct methylation sites that differentiate between the different meningiomas. And then finally, using the machine learning model for predictive classification based on the extracted information we obtain from radiomic features and methylation status of key genes.
Diving into the methodology of our research proposal, for our data collection protocol, we plan to obtain data from a multi-center study to improve data heterogeneity. We will recruit patients diagnosed with meningioma by certified neurologists and obtain the 3 MRI scans of their brain that we discussed, the T1-weighted, T2-weighted, and FLAIR scans, as well as epigenomic data on the methylation status of their genome. And patients with incomplete data will be excluded from this study to ensure our machine learning models we develop are accurate and robust. Following this, for the segmentation aspect of our project, radiologists would first manually annotate ground truth labels for the tumor location, which would be used to compare predictions to during training. A modified 3D unit convolutional neural network, which is a specific type of machine learning algorithm, will be trained for tumor segmentation using a 70-30 train test split of data. And it'll be validated using the dice score metric, which is a measure of overlap between predictions and ground truth. Next, 200 common 2D radiomic features will be extracted from the tumor, including characteristics like tumor size, shape, contrast, and many more. And then finally, statistical analysis will help identify the most important features distinguishing between the different grades, which we can then integrate with clinical or genomic data.
So in terms of the methylation analysis, we will use sodium bisulfate conversion and sequencing, which is the current gold standard to detect DNA methylation in samples. Following this, we will also use statistical analyses to compare key genes differentiating tumor grades, using techniques such as unsupervised clustering. A brief literature search yielded us the following 9 genes, which would be the initial focus of our investigation.
So for the final stage of our proposal, which is the radiogenomic prediction, we first propose a resonant machine learning model to predict the methylation status of key genes using these radiomic features in a similar algorithm to existing papers that predict the methylation of the MGMT promoter gene in glioblastoma. Second, a classification machine learning model will take the radiomic and genomic features that we've identified and train it itself to classify the patient as benign, atypical, or anaplastic meningioma. All in all, we envision our classification to be rapid and effective, and our final pipeline would be something like follows. A suspected patient gets a referral from a doctor, and they'd get MRI input images, from which automated radiomic feature extraction would occur. Next, a prediction on the methylation status of key genes would be derived, and then finally, our radiogenomic model would grade the tumor, leading to a rapid transition towards treatment and management. Our research proposal also holds numerous additional applications outside of meningioma grading, many of which have been supported by existing literature. Having a segmentation algorithm at the start can also assist with the machine learning-based diagnosis of a tumor itself compared to healthy patients. The radiogenomic algorithms can be used for other cancers as well. And the radiomic features we extract can also be used to predict survival or recurrence of tumors.
In conclusion, our goal is to increase diagnostic rates, build upon tumor knowledge, and increase patient survival through this novel machine learning model to classify meningioma by grade. The proposed model of ours is standardized, cost-effective, time-effective, non-invasive, and individualized, and we believe it can contribute to significantly improving the meningioma clinical workflow, giving hope to patients like Andrew. Thank you so much for listening, and we'd be happy to take any questions you might have.
Thank you very much. That was great. I will now pass the floor over to the judges for any questions that you might have.
Hello, it's Adriana here. Oh, my connection is unstable. I think I have to go off video too. I'm sorry. Are you still able to hear me okay?
Yeah, we can hear you.
Oh, that's good. Okay. Well, thank you so much, Dhruv and Gurdeep, for preparing and presenting this research idea to address these problems and questions around atypical meningioma. Your letter of intent, it was very nicely structured and written for starters. And I also really enjoyed your presentation and congratulate you both for coming up with this proposal. So in a nutshell, then, I understand you're trying to use cancer imaging features to predict if these tumors have genetic alterations that relate to higher grade, and then to get imaging biomarkers to replace biopsy. So your target is for unresectable tumors mainly, is that right? Or for any tumor?
So the idea is that it can be converted for any tumor, and we just have to eliminate the use of biopsy in the first place to sort of decrease the invasiveness and the cost of the overall grading procedure.
I see. So machine learning can predict the grade and maybe spare patients surgery. What sample size would you need to develop a reliable paradigm for this?
So in terms of machine learning projects, typically the data sets are upwards of 1,000 patients. We're hoping that with the incorporation of a multi-center study, if we can incorporate, say, 5 different clinics or centers, we might be able to have 200 per, and then we'd reach a rough data set of 1,000. But with machine learning, the more data you have, the more heterogeneous it becomes, and the stronger your machine learning model will be.
Okay, well, thank you very much.
Thank you for your
Thanks
question.
oh, sorry. Thanks, guys, for your presentation. I thought that was very well done. I had a clarification about your methodology. So you very rightly mentioned the heterogeneity that can be found within these tumors, and you did a nice job of mapping that both visually and with your words. But one of the questions I have is, if you're looking at things like, if the machine learning will evaluate things like, the textural analysis that you commented on, I suppose you would need entire total resection of real pathology, like the real tumors, to compare the imaging to the actual tissue to.
So in terms of data sets in meningioma, at least when I've investigated data sets in glioblastoma, a lot of those features have actually been derived just from the MRI images. They're provided in clinical data set. I suppose when collecting new images for the training set to compare the features extracted to ground truths, we might need to conduct some resections. But because that's currently in the clinical workflow, it won't add an extra step. It'd just be continuing with the existing workflow to collect the data and then removing that step from the workflow in future when this application could be applied.
I suppose that okay, so if you have features that are identified on your machine learning as potentially areas that are higher grade, I guess, how are you going to confirm that those are accurate? Right, the machine isn't going to know whether it's accurate or not. You need some sort of ground truth, as you correctly put it, to compare that to. How do you see yourselves doing that?
So the idea is that in the data set, we'd collect patients directly that have undergone the clinical workflow. So when they currently go to the clinic, they would be undergoing the biopsy procedures, and then we'd get the information of what grade the pathology lab would get, as well as the MRIs annotated manually. And then from that, we develop our model to sort of predict these features so that in the future, it wouldn't be necessary.
Okay, excellent, thank you.
Oh yeah, I would agree with what Adam said. I mean, you still need the gold standard, which is the pathology. Without that, you can't really say for sure that your project will come up with a meaningful answer. My one question is you picked, I think, what you called representative sequences, T1, T2, and FLAIR. How did you come up with those?
Do you want to answer that, or do you want me to take it?
Yeah, you can take that one.
Okay, so basically, when we were looking at existing literature, we analyzed different MRI scans that are often used for the diagnosis of brain tumor, and we found that they each have different characteristics. So for example, FLAIR MRIs often highlight abnormalities, and then CSF flow is different between T1 weighted, T2 weighted. So we thought that our machine learning models would be most robust and most effective if we incorporated a wide variety of features, and we felt that using all 3 different types of MRIs would help synthesize the most amount of features to use.
Is there one sequence that you may have missed that might be actually the best for meningioma in your reading?
I'm not sure. Perhaps there's one. I know there are certain scans like the ADCP-val or the K-trend scans, but in our research, we didn't directly encounter those as being superior, but I'm not sure if you have a suggestion.
So I would suggest you add a contrast medium agent called gadolinium. Meningiomas can actually sometimes not even be seen without the addition of gadolinium if they're particularly small. And that's kind of standard of care for any patient that's got a brain tumor, especially a meningioma. So I would add that T1 plus GAD, T2, and FLAIR would probably cover it. Okay, thank you.
Okay.
Thank you.
Thanks so much, guys, and thanks so much to the judges for the questions. We will now move forward to the team MacMaster. And Wenjun, you should have access now to present. Oh, there you are. Perfect. So whenever you're set, we'll do the same strategy of the 1 minute.
Okay, are you able to hear us?
Yeah, we can. Thank you.
Okay, cool. So I can start whenever. Okay, hello everyone. My name is Wenjin. I'm here with my colleagues Kevin and Wassam, and we're second years from the MacMaster University. And we would like to thank the Brain Tumor Foundation of Canada for giving us this opportunity to present our proposed research on the use of nanoparticle carriers for improved blood-brain barrier drug delivery and reduced cytotoxicity.
Right, so just giving an overview of our presentation, we're going to begin with an introduction where we introduce the case study, our background, and our research methodology, as well as our hypothesis. We're going to be talking about our aims and which methodologies we're going to be using to achieve these aims, as well as the statistical analysis. And finally, we're going to be discussing our rationale.
So just to give a brief recap of the case study mentioned, it was about a 55-year-old software engineer by the name of Andrew. He underwent numerous scans, and through a craniotomy, it was found that he had an atypical meningioma. So from here, what Andrew can do is he can undergo a surgical excision of the tumor, and then radiotherapy in conjunction with chemotherapy would likely follow. But the main issue with this is that most cytotoxic drugs run too high of a risk of side effects, especially when used long-term. And this combined with the blood-brain barrier's high selectivity could prove to be quite an issue. And that's where our technology of polymeric nanoparticle carriers and interferon alpha comes in. So recombinant interferon alpha is observed to hinder growth of cultured human meningioma cell lines in vitro. And also previously, many nanomaterials have been constructed to modify the surface of blood-brain barrier targeting ligands, such as those seen in the diagram to the right. So our technology will specifically use polycaprolactone, which is a popular polymeric nanoparticle. And by functionalizing this with octreotide, we can control the surface properties and direct it to the tumor site. And here's an example of the chemical structure of PCL. So we hypothesized that the use of PCL-MPs surface functionalized with octreotide to improve the diffusion rate of IFN-alpha through the blood-brain barrier, as well as reducing cytotoxicity, will then also allow for improved drug efficacy. So with this study, we aim to conjugate an octreotide to PCL, which would in turn promote the endothelial cell receptors recognition. And this results in transcytosis, which increases the aqueous solubility and diffusion rate, and by extension would allow more plasma to cross the blood-brain barrier and reach the brain. And for our second aim, we aim to lower the cytotoxicity of IFN-alpha by using PCL in conjunction with the surface conjugation method, which improves drug loading, specific biodistribution, and would selectively target the brain tumor cells.
So moving on to our methods, we're going to begin by discussing the formation of the tri-block polymer, followed by the formation of the microsphere. We're going to be talking about how we're going to be analyzing our microspheres by determining its cytotoxicity in vitro, as well as its efficacy in vivo with mice, before discussing the statistical analysis we're going to be used to evaluate the significance of our data. So starting with the synthesis of the polymer, we're going to be forming a tri-block polymer, or a polymer that's composed of 3 repeating units. So we're going to be using ring opening polymerization using the caprolactone ring you can see on the screen. And we're going to be opening onto either side of the polyethylene glycol, or PEG. And this is going to form a PCl, PEG, PCl tri-block polymer. Now moving on to the formation of the microspheres, we're going to be using multiple emulsion. So essentially with multiple emulsion, what happens is that we're going to be using a methanol oil phase as well as an inner aqueous phase. And we're going to be stirring these together to form nanoparticles inside of the oil phase. The MEO phase is going to be composed of octreotide, methanol, and our TB or tri-block copolymer, whereas the inner aqueous phase is going to be composed of the IFN-alpha diluted in a saline solution, as well as HSA, which is human serum albumin, which is co-encapsulated in order to protect against degradation. We're then going to be repeating this process again with another aqueous phase that contains water and a polaxomer surfactant, which is going to be helping with the dispersion of the water in order to form that second emulsion, hence multiple emulsion. These microspheres can then be freeze-dried and used later. So starting with the analysis of the nanoparticles, we're going to be testing it for size, encapsulation efficiency, stability, structural integrity, and release. We're then going to be evaluating the cytotoxicity using a hepatic cell line on a 96-well tissue culture plate. Cell viability is going to be used by colorimetry analysis, and how we're going to be doing that is we're going to be measuring the metabolism of the cells with a tetrazonium salt to see whether they're actually metabolizing the salt and then the color changes, or whether they don't metabolize the salt. And we're going to be determining the viability by seeing if it's above 50% or below 50%. Finally, we're going to be using an in vivo analysis with in-bolus injections of 8-week-old nude male mice, which are going to be injected with a meningioma cell line. And then we're going to be measuring the reduction in tumor size upon intravenous injections of the polymeric nanoparticle using bioluminescence imaging and caliper measurements.
So just to affirm our experiments, we hypothesize that the results will be that through conjugating octreotide onto a PCL will allow for more selective targeting of the meningioma, and this is due to the high surface binding to meningioma cell lines, and in contrast to low surface binding to our arachnoidal cell lines. And this would in turn promote blood-brain barrier penetration and lower cytotoxicity.
So moving on to statistical analysis. So as Sam has mentioned, we'll be having a control group and a treatment group for the mice, and we'll be measuring the size of tumor reduction in millimeters cubed. And from the data collected, we'll then be using the data for a paired t-test, where we put the data through Microsoft Excel, and then the Excel formula will provide us with a t-test p-value. And for this t-test, our null hypothesis is that there's no statistically significant difference between the means of the treatment group and control group. And of course, our alternative hypothesis is that there is a statistically significant difference. If the p-value is less than 0.05, then we can reject the null hypothesis, and we can accept that there is a 90% confidence level that there is a statistically significant difference between the means of the groups. However, if the p-value is larger than 0.05, then we have to accept the null hypothesis, and there is no statistically significant difference there. So now going to our rationale, we have considered many factors in affecting the efficacy and the usability of our treatment methodology. So we have chosen PCL for its high biocompatibility and slow degradability. And for our copolymer, which is the PEG PCL copolymer products, it has been showing studies that these products, much like the nanoparticle that we proposed for using, have no concerns with toxic side effects. In addition, we have decided to functionalize our nanoparticles with octreotide. This is because octreotide is an analog to the stomatatin, which has its receptor, which is highly expressed in meningogenomic tumors. So by using this, we're able to target specifically those receptors on the tumor cells. And finally, we have chosen IFN alpha for a drug because it has been shown to have strong tumor inhibitory effects. And here is our references.
And once again, we'd like to thank the Brain Tumor Foundation for this opportunity. And thank you for your time and listening to our presentation.
Thank you very much. That was great. Judges, I'll open the floor to you.
Let's see. Hi there. You're able to hear me okay?
Yeah.
Okay. Hi, Wenjun, Kevin, and Wissam. I have to turn off my video again. I'm really sorry. There. Okay. So yeah, thank you so much for presenting your research idea this morning. I really enjoyed reading over your letter of intent too, and then hearing your description of the project just now. So as I understand, you want to see if you can direct a therapeutic drug, the alpha interferon, to the tumor target using an octreotide-loaded microparticle. Is that right? Okay.
Yes.
I think you have a very nice model of in vitro and then translated to an in vivo mouse framework there. And it seems very logical to me, but I wonder, I have a couple of questions. Why not give the octreotide conjugated to interferon itself, since most meningioma, in my understanding, are actually outside of the blood-brain barrier? They're arising from arachnoidal cap cells, and they're not really primary brain tumors that are limited by the blood-brain barrier in the way a glioblastoma would be. And your nanospheres are designed to pass the blood-brain barrier, and you're functionalizing them to target the tumor with the octreotide. But do you actually need to take that step of using this particular carrier? So that was one question. And my other question was, since there are somatostatin receptors in many other organs and tissues, could you anticipate any off-target effects of this type of approach?
Right. So addressing the first question regarding the blood-brain barrier, part of the study would also be that IFN alpha isn't only for meningioma; it can be used in other tumors as well. So maybe by kind of passing the blood-brain barrier, it will allow us to extend this study into other areas like glioblastoma and other tumors. And regarding the other question, somatostatin receptors are, it's especially overexpressed in meningioma cell lines. So so that will help with the selectivity. With any other drug, you also run the risk of perhaps killing other cells or healthy cells, but we're going to be using this study in order to assess the selectivity and whether the efficacy of the drug to make sure that it can be used in human trials if it comes to that.
Yes, and adding on to that, because the receptors are widely expressed in these tumors, we think that even though there is still a risk that it could be mistargeted into other with proper injection of the drug, and we think that it will be better than just without the functionalization of triotide in terms of specific targeting.
Okay, thank you very much.
If this project was successful and was eventually used in humans, do you guys envision this as a complementary treatment to surgery, something that would be used with or after surgery instead of radiation? Where do you see this fitting in the treatment scheme for these patients?
Right. So generally, chemotherapy is not the primary treatment for meningioma. The gold standard would obviously be resection. And then chemotherapy is used if you can't fully resect it or if you see a regrowth of the tumor. So we feel that it would be more it would be more plausible to use as an adjuvant therapy. And then further studies on top of this would also maybe allow it to go into that more primary treatment in cases where resection isn't possible or there are other complications.
What might be some adverse effects from this treatment? Are there any that you've considered, or do you think it's a pretty innocuous treatment?
Yeah, so like as we kind of talked about, I mean, there's always the potential, because somatostatin is also expressed in other organs, the potential to kill healthy cells as well, which you run with any chemotherapeutic treatment. So that would definitely be our biggest concern. And by analyzing the cell death, we can see what's that optimum dose of fish, like what is the optimum dose to ensure we're killing the most meningioma cells or the most brain tumor cells, whereas and simultaneously killing the least number of healthy cells. So that would be our biggest concern going into the project.
So to your knowledge, then, I'm assuming the interferon is the anti-tumor component. Is that correct?
Yes.
Does interferon have any other side effects on patients that it's been used on in the past? You're talking more about a specificity issue, but in general.
In general, I think through our research, we've only found preliminary testing for the interferon. So I don't think we found anything regarding clinical trials on patients. So that's why we're also proposing the use of it, just to test the use of the efficacy of the drug along with our nanoparticle.
I mean, I think if you look back, interferon at one point in history was thought to be going to be a wonder treatment for things, and I think it didn't really turn out to be that way. But I mean, it's still, the way you're using it, maybe it will. That's good. Thanks for the presentation.
Thank you very much. We will now move forward to Western University. And Jackie, you should have access to share your presentation. Thank you. Whenever you're.
All right. So good morning, everyone. My name is Rafe, and my partner is Jackie V, and we're both representing Western University. Unfortunately, Jackie has COVID, so we won't be able to present today, but he's here on the call with us, and we're going to try to make the best of the situation. So moving forward, our presentation today is lock-on missiles. using antibody conjugated dual package stable nucleic acid lipid particles to deliver microRNA and small molecule inhibitors against GATA4 expressing atypical meningiomas. Meningiomas are the second most common group of intracranial tumors and are characterized by a high mitotic activity with an increased cell proliferation rate, hypercellularity, which is the presence of abnormally excessive amount of cells, and the sheeting phenomena, in which the cells organize themselves into an unusually ordered formation, which is very distinct and characteristic of atypical meningiomas. Transcription factor proteins control how other proteins are produced from DNA. Recent research has identified that GATA4, a transcription factor protein, is excessively overproduced in higher grade atypical meningiomas and therefore represents a novel therapeutic target. The oncogenic ability, or the ability to induce the risk of tumor formation of GATA4, stems from its involvement as a regulator in the production of other key oncogenes such as cyclin D1. GATA4 functions by inhibiting the production of microRNA 497, which inhibits cyclin D1 production. Essentially, it stops the microRNA from blocking production of cyclin D1, resulting in an increase in tumor size. Furthermore, other research is starting to suggest that microRNA 497 levels are an effective indirect measurement for the activity level of GATA4 and hence can be used as a biomarker. Before moving forward, I will quickly go over the concept of a targeted dual package stable nucleic acid lipid particle, or a SNELP, which is what I'm going to be calling it from now on, and how they can be used to target specific tumors. Firstly, many current chemotherapeutics come in the form of small molecule inhibitors, which are chemicals that interfere with the functioning of proteins that are associated with cancer. Secondly, an emerging field of cancer treatment comes in the form of RNA interference, which employs the use of microRNAs that bind to and prevent these oncogenes from being translated into active oncogenic proteins. Thirdly, a single SNELP can be loaded with both the microRNA and the small molecule inhibitor and use these SNELPs' lipophilic properties to permit passage of the drugs through the restrictive blood-brain barrier and access tumors. Finally, to ensure targeting specificity, the SNELP can be conjugated with antibodies that are specific against the specific type of tumor you may want to target. Now that we've kind of established how and what dual packaged antibody conjugated SNELPs are, let's discuss some of the current issues surrounding the treatment of atypical meningiomas. So as we saw with Andrew in the case study, the most common course of treatment is surgery to physically remove or reduce the tumor mass. However, 1/3 of meningiomas are biologically aggressive or surgically inaccessible, and atypical meningiomas are known to have a high rate of recurrence, which would then necessitate another round of surgery, which can be extremely debilitating for patients. Therefore, there exists a need to find alternative targeted therapies to help those affected. So this is where our previous discussion about dual package antibody conjugated SNELPs comes into play. It will allow us to reach these surgically inaccessible atypical meningiomas and also suppress the reoccurrence of tumors after surgery. So what would a dual packaged antibody SNELP construct specific to GATA4 look like? Firstly, a previous study has shown that the small molecule inhibitor 1,3-benzodioxyl-5-O-methyl can maximally interfere with GATA4's ability to interact with DNA. This is the small molecule inhibitor that we will be packaging in the SNELP. Secondly, previous research has also shown that the microRNA 26B is the main microRNA that inhibits the translation of GATA4. Thus, we propose a dual packaging of both the 1,3-benzodioxyl-5-O-methyl and the microRNA 26B. Targeting specificity will be achieved through antibody conjugation, specifically with the anti-SSTR2 antibody, as shown in previous research, as being specific to target atypical meningioma. So from this overall mechanism, we hypothesized two major results. Using mice expressing atypical meningiomas, we hypothesized that both the single packaged and dual packaged antibody conjugated SNELP constructs will reduce the total tumor volume, and we'll confirm this by imaging using CT scans and compare these results to control mice. So our first aim would be to test this hypothesis using 3 treatment groups. The first one being mice administered the dual package SNELP. The second group being mice administered the single package or microRNA containing only SNELP construct. and a control group of mice administered a saline placebo. We predict that the dual package SNALP and single package SNALP construct administered mice to have a significantly reduced tumor size compared to the control mice, and that the dual package SNALP construct administered mice will have the greatest overall reduction in tumor size. So to assess the specificity of our SNALP constructs to atypical meningiomas, we will use comparisons of cardiac hypertrophy. Well, why cardiac hypertrophy, you may ask? Because GATA4 also acts on cardiac muscle cells to increase hypertrophy or muscle growth. Hence, off-target inhibition of GATA4 would reduce cardiomyocyte hypertrophy. Therefore, we will compare cardiac hypertrophy in mice injected with the dual package SNALP construct conjugated to the anti-SSTR2 antibody with mice that are injected with a dual package SNALP that it is not conjugated to any antibody. And we predict that mice injected with antibody conjugated SNALPs will have significantly increased cardiac hypertrophy compared to the non-conjugated SNALPs. Our third sub-experiment will seek to confirm the validity of microRNA 497 as a potential circulating biomarker for atypical meningiomas through the quantification of the microRNA levels using RT-PCR. We expect the quantity of the microRNA to be decreased the most in mice injected with the dual package SNALP constructs. So the current treatment paradigms recommend adjuvant radiotherapy following surgery to remove grade 2 tumors. However, radiotherapy is fraught with problems, with toxicity rates which extend up to 16.7% and a myriad of unwanted side effects such as cerebral necrosis and a 12-fold increase in the risk of stroke. In contrast, our proposed study involves a novel therapy which can replace radiotherapy as a post-surgical treatment with less side effects than conventional radiotherapy. If our results are consistent with our hypotheses, then there will be a few points of therapeutic relevance. Firstly, we can now use microRNA 497 levels as a reliable biomarker for the detection of tumor reoccurrence and to ensure the efficacy of our SNALP treatment. Secondly, a dual packaging of both the microRNA and the chemotherapeutic drug can far better suppress atypical meningioma compared to giving either of these by themselves. Thirdly, the increased specificity confirmed by the conjugated antibodies minimizes the side effects by reducing off-target actions by the SNALPs. And finally, our proposal will lay the foundation for future clinical trials. So for Andrew, what this means is management of his atypical meningioma can be achieved through both biochemical observations of his microRNA 497 RT-PCR levels and through active suppression of his meningioma using our SNALP construct. Returning to the idea of clinical trials for Andrew and his family, we could test the dual packaged antibody conjugated SNALPs in humans to confirm their safety, determine dosing, and gauge their efficacy. One hypothetical design of such a study can be the one depicted on our slide, with treatment groups being patients with atypical meningiomas being treated with only surgery as our control group, and patients treated with either the single or dual package SNALPs following surgery, with the greatest predicted reduction in tumor size occurring in patients treated with the dual package SNALPs. So thank you everyone for listening to our presentation, and we open the floor for questions.
Thank you very much. I'll open the floor to the judges.
Well, hi, Rafe. It's Adriana here. I'm sorry to hear about Jackie coming down with COVID. That's too bad. But I certainly thank you for presenting this really interesting research idea. And I enjoyed reading over your team's letter of intent too. It was really very clear and nicely written up, and it was nice to hear the details in your presentation just now. So you want to deliver a small molecule inhibitor against GATA4, and that's oncogenic, and it has a surrogate marker that you can use to measure to see if therapy is working.
Yes.
And you want to package it in particles that can cross the blood-brain barrier and target meningioma specifically with the antibody against a somatostatin receptor. Is that right? That's it in a nutshell, right?
Yes, I mean, it's a big project, but yeah, like in a nutshell, that's what our idea is.
Okay, so again, I have the same question for your project. You know, do you really need to worry about getting your therapeutic agent to cross the blood-brain barrier, for one? I guess in invasive meningioma where there's actual brain invasion, it might be helpful that way. Or for the inaccessible tumors maybe, or I don't know, residual or recurrent tumors. But yeah, and again, I just worry about the off-target effects because, you know, targeting somatostatin receptors, I think, has some downsides.
Jack, do you want to take this one away?
I'll try not to cough as I say that. So for the
Oh,
blood
poor
brain
you.
barrier,
Oh no.
for the antibodies, the anti-SS or TR2 antibodies, they still have to be embedded in the SNPs in order for them to like work effectively for the package. So even if crossing the blood-brain barrier isn't that much of an issue, just having a packaged lipo nanoparticle for the microRNA and the chemotherapy to begin, and to have the, I guess, the lipid bilayer just for the antibodies to be embedded in, to enable the entire construct to work as a functioning vector. That would be the basis for having a SNP. And can you please repeat the second question?
No, I think that that's okay. I just wondered what the off-target effects of this might be, you know, if you're using these small molecule inhibitors against the tumor, what would the effect of the small molecule inhibitors be against, say, functioning endocrinological tissue or the gut or places where somatostatin receptors are? You know, just the off-target effects, that's all.
So like to answer that question, essentially that's like the purpose of our second experiment. So with these specific microRNA that we're using, we found a previous study that kind of showed that it was able to reduce GATA4 expression in cardiomyocytes and then reduce hypertrophy. So that's why we proposed that like second sub-experiment in which we're kind of looking for like an inhibition of cardiac myocyte hypertrophy. So that's kind of like we took that risk into account, and which is why we have that second experiment to kind of test and see. Obviously, this is like very hypothetical, very theoretical. This is like years worth of research to make this come alive, but we did account for that with that secondary experiment.
Also, to add on to that, if we didn't conjugate to an antibody against a somatostatin receptor, and we were just to give the small molecule inhibitor as an IV injection bolus, regardless through circulation, it would probably end up in the endocrinological tissues or in the gut. So we thought that we feel that since meningiomas and other cancers usually overexpress the somatostatin receptor to such a great extent, the side effects that may occur with our treatment now would be, compared to if it was just an IV injection bolus, be much less than without the conjugation to the antibodies.
Okay, well, yeah, thank you. Yeah, that answered those questions, and especially the cardiomyocyte and cardiac hypertrophy aspect of it. That really wasn't clear in the letter of intent, but it makes more sense now. Thank you.
Yeah, you guys have done a great job answering the questions. You've answered the ones I was going to ask already. I guess I can, I saw that you had sort of a quick proposal for a potential clinical trial design in the future, and I think that was wise to include. You know, I suppose, are you envisioning this as, I suppose it would depend on the response, but do you envision this as something to replace radiation, replace surgery, or as a complement to either one of them? Like, where do you think this fits in?
So essentially, like to answer your question of where it fits in, I guess, like in both. So essentially what we kind of, or what we're proposing is that it's used after surgery. So it would replace the role of adjuvant radiotherapy, as has much, or we expect it to have much less side effects than radiotherapy. And then we also mentioned earlier in our presentation that I think 1/3 or a small or a decent percentage are surgically inaccessible. So for people that have meningioma that's surgically inaccessible, this could be a way to reduce that and attack those tumor cells if they can't have surgery in the first place.
Okay, thank you. Very clear.
So in your letter of intent, recent research has shown that GATA4 is overexpressed in higher grade meningioma. So is that, just for my education, is it overexpressed in all patients with higher grade meningioma? Like if there were 10 patients with an atypical meningioma standing in front of me, how many of them would have overexpressed GATA4? All of them, or one of them, or?
Yeah, you want to take this one, or you want me to take it?
And take it. So for that research paper, I would feel that GATA4 generally just as a transcription factor that's involved in cell proliferation for meningiomas that range from benign to malignant. Usually there is some initial amount of growth that occurs irregularly. So to get to a point of a, I guess, benign tumor mass being established, there has to be some upregulation of GATA4. For our research proposal at the level of an atypical meningioma, the levels of GATA4 that are expressed are sufficiently high enough, according to the paper, to be targeted as a potential therapy target.
Thank you. And my final point was related to what Adam was asking you. I mean, I think you guys are a little hard on radiation therapy. Radiation therapy can be quite effective in patients with brain tumor. And I think if you were to do a clinical trial as you propose, you would need a treatment arm that would have radiation therapy in it, because it is kind of standard care for higher grade meningiomas now. I'm not saying that your compound or your mechanism couldn't potentially replace it, but you would still need to demonstrate that. Thank you.
I mean, that makes complete sense, right? Like if we're suggesting a brand new therapy, we need to compare it with the gold standard to see if it's better, because if it's not, then there's no point in doing it.
Well said.
Thank you very much, guys. We will move on to the next group. And Jackie, we hope that you're feeling better soon. So the next group is McGill, and Nevia, I believe that you should have access already to share your presentation. Thank you. Whenever you're set.
Hi, I'm Nivea. And I'm Noor. And we're MDCM candidates from McGill University. And we're more than pleased to present to you our mobile health app for post-meningioma resection. It's called Good Day, Bad Day. So first, we'd like to acknowledge the Pam and Rolando Del Maestro family and Brain Tumor Foundation of Canada for giving us this opportunity to present. And just an overview of the case, the patient Andrew presents with forgetfulness, balance problems, and a moderate headache. He goes through imaging, and we find there's a large lesion in the left ventricle. Then he goes through a craniotomy, and the brain lesion is resected. Now, Andrew and his family meet with the neurosurgeon, neuro-oncologist, and radiation oncologist to discuss the next steps. So just a brief background on meningioma. It's mostly benign and it's the most common brain tumor, but it could be resected. But post-resection, there is shown to be reduction of the quality of life, socially, physically, and emotionally. And there have also been patients with symptoms of depression or anxiety, as well as patients experience pain and social isolation. So the intervention we thought of is a mobile health app. It's called Good Day and Bad Day. And what we find is that normally it's difficult to accurately quantify this distress and get a general practitioner's input. And we feel that another problem would be that there's a loss of follow-up and lack of accessibility. So we believe there's a need for a standardized and validated tool to help measure emotional distress, which is why we have Good Day, Bad Day, which would send periodically a questionnaire to the patients, and it will be accessible to the general practitioner as well, but it will be confidential. It would assess the mental health and the emotions that these patients will be feeling. the hypothesis would be to see if the implementation of this app with the standardized questionnaire as a screening tool will help to improve the quality of life of these patients post-surgery. So we have three main objectives for this research effort. Firstly, we want to determine if Good Day, Bad Day will improve the quality of life in patients post-surgery. More specifically, we want to examine if the mobile health app will alleviate emotional distress and improve their mental health. And then we will study whether the mobile health app provides an easy accessibility to the majority of patients. So our research is mainly qualitative, and we base our methodology on two main theoretical framework. Firstly, it's the patient and public involvement in the health research process, so the PPI methodology, which basically consists of undergoing research and including patients and the public, so that it's being carried out with or by members of the public rather than to, about, or for them. To do so, we will use the GRIP2 guidelines that are shown in this table throughout the research. The second theoretical framework that we base our qualitative research is the experience-based co-design methodology, so the EBCD, which is an approach that actively involves patients as well as staff members in quality improvement in the healthcare system through narrative-based approaches. Because we will use those theoretical framework to co-design a questionnaire that will be sent in the mobile health app. So now I'm just gonna do a step-by-step process of the research effort. So firstly, we want to develop the questionnaire. So the first version will be developed by the research team. It will be evidence-based, and as such, we will perform a literature review to develop the screening tool. Once this screening tool is developed, we will try to do a second version, but with the it will be focus group based. So this we will firstly need to recruit key stakeholders, which include patients who had surgically resected meningiomas, families and caregivers of the patients, and healthcare professionals that we all be it will all be organized in focus group, and they will give their assessment of the developed tool through meetings. Once the meetings are done, they will be recorded, transcribed, and then the research team will thematically analyze this. And so how does it work? It basically, with the qualitative data that we will have through the transcription, we will code the transcription and then extract the themes. With the themes extracted, the research team will include the recommendations done by the focus groups. And then we will have a final meeting with the research, with the focus groups, and then they will give us their last opinions on the third version of the research. So finally, we will assess the validity of the questionnaire, firstly by reviewing the third version by a group of experts in mental health, which can include psychiatrists, therapists, psychologists, social workers, and then to assess the measurement capabilities of the tool, we will hold a small pilot test that will include 5 to 10 patients and their physician and healthcare provider, just to collect a few samples of answer and see if the answers make sense and if it's easy to use. Once we're satisfied with the pilot test and that we made the last amendments, we will hold a randomized controlled trial that will include 50 patients. The first group will be the control trial and will be assigned to the traditional approach of screening for mental health, which is self-referrals or the general practitioner judgment referrals. And then the other half will use our developed screening application, as well as being followed by a GP. They will be observed for 5 years, and then we will assess for a number of outcomes that are listed, such as acceptability, accessibility, compliance, quality of life. Finally, the hypothesized results is that we believe that the app will improve quality of life post-surgery and reduce emotional distress for a cancer patient, because other apps have shown a positive impact. And finally, the goal of this app would be to ensure that there is a follow-up of patients, and very rapidly, because this would be answering a survey on your phone, and it's a matter of a few minutes in their hands. So this would help post-surgery patients to cope with their emotions. And always we emphasize there are physical limitations post-brain tumor or surgery, but we need to also prioritize mental health for these patients. And finally, it'll help these patients heal as a whole, because it will be with them throughout their journey in fighting against a brain tumor and recovering from the mental health scars that it may leave behind, whether it is a good day or a bad day. Thank you so much for listening to us.
Thank you very much. I'll open the floor to the judges.
Let's see. Am I off mute here? Yeah. Okay. Hi, Nevia and Noor. It's Adriana here, off camera. I'm so sorry. But I really wanted to thank you for sharing your research idea with us. It is really true that some patients with meningioma experience a pretty significant impact on their quality of life. And so it was nice to see a quality of life project within the spectrum of proposals that we've received for this round of competition. So I congratulate you for that. I think that's great. And your concept of a mobile app is an interesting one. A question that came to mind for me relates to privacy and confidentiality, and who has access to the personal health information and the information that the questionnaires are going to generate, and how it's kept safe. And also, in a semi-related way, maybe how that information could be potentially useful as data for ongoing research. So those were questions that just came immediately to mind.
We were initially thinking of having the data being only available to the general practitioner who would follow the patient. So because in that sense, I feel like it would prevent a patient from lying about how they feel, because they would know that it is only me and my general practitioner who would know this and no one else. And we were thinking of having it like encrypted if possible, just like how, let's say we're doing research for doctors and stuff, it's all data that's in the system like Oasis, but we would have access to it in terms of research, and the patients would have to consent to that.
Okay, thank you for that. And then you mentioned in your presentation, also in your letter of intent, an ongoing review of patients using this app over 5 years. And then what happens at 5 years?
So the 5 years is for the research. Obviously, they will continue to be followed by their general practitioner. But after 5 years, like after the randomized controlled trials, so right now we're only targeting so patients, but we know also that they can also be cognitively impaired with the surgery. So our goal, the next step of the research, so after the 5 years, is just to enlarge our randomized controlled trial and include caregivers more specifically, to see if the app can be used by caregivers that have that need to take care of patients, and that the mental health of patients that are so cognitively affected by the surgery is also assessed. And yeah, so it will just be to increase the research effort to see the preliminary result. If with cognitively intact patients, it's a positive result, just to
expand it.
Exactly.
Okay, thank you very much.
You're
welcome. I would echo that. I think the focus on quality of life is a good idea, and you've got an interesting approach to it. in a world where we have an increasing number of apps, many of which are sort of becoming more personalized and integrated with healthcare, did you guys do much in the way of competitive analysis to see what other apps were out there and what yours would offer in addition? And then the second piece is, do you have an idea, just a ballpark number of what it would cost to run a randomized control trial with 5-year follow-up? So 2 questions there.
Okay, I could take on the first one. We did do research and it was really shocking to us, but we did find apps, but it was mostly like breast cancer and stuff. And it was not even in North America. It was mostly like in China or like elsewhere in the world. So that's where we were like, why is this not like, especially I feel like in North America, we use our phones a lot. We're known for, let's say, the Hollywood industry. So like, that's why we were like, why is this not a thing? And that's why we decided to go on with this project. And I don't know if you want to take the second one because I'm not sure.
So for the second one, honestly, we have not, we have no idea about the cost, but yeah. But for the first question, also, as she said, it's mainly very targeted about like cancer types, and there's none to address neurological tumors, and none for like specifically screening for psychological effects, and also they're very more so of group therapy, let's say, and not really like our idea is more of information that will go directly to the healthcare providers, and we have not found anything of that would look similar to that.
Yeah, I think that's a unique aspect of it too. So well done. And just final point, I think it's great to say I don't know. So the fact that you don't know that the cost of a trial, that's okay. That's good to say, and that's something you could look into if it got to that phase. So that's fair.
What I'm a little unclear on, and maybe you said this and I missed it, but so these people would have a mobile device with an app on it. Is that correct?
Mm-hmm.
So then do they tap on the app and then feed back to the headquarters, or?
It would be like an app that would send you a notification maybe like once in a while, once a week, or like in a timely manner that would keep you in check. Because I feel like, yes, there might be family members, but they're not always there asking you, are you okay? Like, is everything okay? Do you feel like you need help? And sometimes family members want to seem strong so that their family members are strong. So I feel like in this way, having an app remind you maybe weekly or daily that it is okay to feel this way and you could reach out for help would be a great idea.
So would the expectation be that when this little chime went off, checking in on you, then you would respond and say, yes, I'm okay, it's a good day, or thank you for reminding me it's a bad day, or something like that? Is that the idea?
If we simplify it, I think that would be the main gist. It would mostly be like writing in like maybe a question box saying how you feel, how your day went. It's not just like rating my days a 5 on 10. It's more about like how you feel, putting your emotions into words.
That's why we're co-designing with many stakeholders to see which questions are geared toward the patients, but also what the healthcare provider wants to know in order to help.
So this app then is kind of in development then.
Yeah, exactly.
Based on feedback from patients and from care providers. And so possible a patient could just say, oh, I think I should tap on my app now and send something, even if I haven't been prompted. That might have that option too, I guess.
Yeah, of course. Yeah, it would have both options, but also a consistent notification, because just to make sure that there's consistency in the screening.
Okay, thank you. That helps.
I had another question just in the context of your presentation. You suggested that patients would be able to access their healthcare provider within a matter of minutes. And that got me to wondering how accessible the healthcare provider on the other side of the app needs to be. I imagine for a, a pilot of 5 to 10 patients, that might not be too bad. But when you expand it to 50 patients and you've got 25 of them on the screening app, it might be hard for a small research team to be responding to all of the patients and responding within minutes. And there's a practicality with that. So that was one question. And then the other was just the demographic of the patients who get high-grade meningiomas. These are maybe older adults particularly. And so what would the patient acceptance for a health app be like? Because younger people are more likely to embrace technology, right? Would this be receptive for an older demographic or a potentially neurologically compromised demographic? And I think you're trying to drive at that with some of your methodology. But I wonder what your thoughts are about it.
I could take on the first question. So basically, like what we meant was not like in minutes to the general practitioner. We meant like in minutes for like the app, like they could just like take the app and start writing. But that is a valid point. I would say that is something that we would think about like when it's a bigger app. But I feel like that would also mean we would have a bigger team. But in reality, it is a very good point, and I feel like we need to reflect on that. And I don't know if you want to take the older patients.
So I think I addressed it a little bit. So for the older or compromise, so of course, that's why also we do the research, because we want to see, like, as much as we really believe in our idea, it's really a good point. The compliance of patients is essential for this to work, as any therapy, any treatment. Patients need to be compliant, and it can be difficult in older patients. So that's why we like the notification aspects also makes it easier, because you just have to, like, once it appears on your screen, you just have to click and try to answer. But that can also be challenging. Maybe they don't have their phone at all times, but that requires education. So that also that will also be an aspect to study, to make sure that general practitioners are also trained in mHealth and eHealth and are able to educate their patient in that, and to see if the education is enough for them to use and if it works. Also, if we see that it doesn't, the compliance is impaired, we can always like use a caregiver that will also receive the app, and it will remind the caregiver to assess the patient's mental health. And
I'll add a last line. I guess if like typing would be a problem, I know there are like technology that exists where, like, let's say doctors could, it would be like verbatim and they would just say it and it would write it down. So maybe that could be an aspect we could include.
Or check, like we can also, there's also an option to make 2 types of questionnaires that's often seen. So a questionnaire that is more open-ended for a longer answer, but if there's an issue with that, there's also checklists, even though it doesn't give as much information. If it's consistent, and if the most important thing is we assess mental health, the more like if it could be just a yes or no, and in patients that need it.
Okay, perfect. Thank you so much. I'm sorry, but we do need to move forward with the next team. So thank you so much for taking the time. The next group is the University of Montreal, and Marianne, you have access already to start your presentation.
Perfect. So hello, everyone. My name is Seychelle, and I'm accompanied with Marianne. We're both undergraduate students at the University of Montreal. We want to start by thanking the Brain Tumor Foundation for this amazing opportunity for undergraduate students. So today our theme is atypical meningioma, and what exactly we want to analyze is the role of prions, proteins, in modulation of tumor-associated macrophages and microglia in atypical meningioma. So first, meningioma are a group of primary brain tumors originating from the meningeal coverings of the brain and the spinal cord. They are classified in 3 grades, where atypical meningioma is grade 2. We have 3 subtypes of atypical meningioma: choroid, clear cell, and atypical. The patient that we were presented in our case study, Andrew. That's the normal patient management. They usually have resection and radiotherapy, but the tumor is not extensively studied as gliomas, for example, and we don't really know about the relationship between the tumor microenvironment and the tumor itself, which makes having a chemotherapy troublesome. But even with that, we know that some gene alterations like NF2 have an impact on the tumor progression. The NF2 gene is located on the 22nd chromosome of the human genome and codes for the tumor suppressor myelin. It was demonstrated that its mutation or deletion can have an impact on the tumor progression. First, the gene mutation increases prion productions in tumors. Prion proteins are normally expressed in every cell of the body. There are two main conformations, the PRPC, which is a normal conformation, and the PRPSC, which is the abnormal one. The abnormal one was known in the 90s to cause prion diseases such as the transmissible spongiform encephalopathies. But we also know that in oncology, recently it has been discovered that the PRPC is also known in oncology for its role in multi-drug resistance, immunomodulation, immune invasion, and stem cell characteristics in tumor cells. So for example, a study has shown that the expression of PRPC increased in the tumor grades in glioma, and where you can see the comparison between grade 1 compared to grade 4, where there's more brown spots in the grade 4, which is the prion expressed. And another study compared normal meninges to grade 1 meningioma have shown a higher expression of the prion protein when the NF2 gene was gone. The second thing is that the NF2 gene may help in the tumor progression in affecting the polarization of tumor-associated macrophages and microglia, or TAMs, as we will call them. It may be involved in the transition from M1, which is more pro-inflammatory or anti-tumoral, to an M2 which is pro-tumoral. So the M1 phenotype is usually associated with the phagocytic activity that we see in the peripheral macrophages, and the microglia are just the macrophages in the cell, in the brain. So the M2 phenotype of the macrophages have a decreased phagocytic activity and help in tumor progression by secreting some factors such as the vascular endothelial growth factor, which help angiogenesis around the tumor. A study showed that the M1 to M2 ratio of the TAMs have an impact on patient survival, where the higher the ratio is, the better the chances of survival. NF2 monosomy, where the mutated form of the gene is gone, has a higher level of the ratio there. So we can see that in the grade 1 meningiomas compared to grade 3, where we know it's anaplastic and because it's very malignant, we can see that is a higher M1 to M2 ratio. Another small study comparing the ratio in 3 patients with choroid meningioma, which is a subtype of atypical meningioma, showed that there's a significantly higher number of M2 TAMs even before relapse compared to a non-recurrent patient. So that study was a very small, only 3 patients. So that means that study would need more further confirmation.
So in this proposal, we wish to study the role of prion proteins in the modulation of TAMs in atypical meningiomas. Our research problems are based on gaps in knowledge. We seek to better understand the role of prions in atypical meningiomas, the tumor microenvironment, and the impact of protumoral cells on anti-tumoral cells in atypical meningiomas. We hypothesize that the tumor cells will release prion proteins, which will induce the polarization of the TAMs from an M1 phenotype to an M2 phenotype. The M2 TAMs will then release more prions, which will suppress the immune activity in M1 TAMs and NK cells, for example. So first, we will do a comparison between the PRP expression of the atypical meningioma subtypes and normal meninges. We would use the mRNA expression with qPCR, the cell lysates for the protein expression intracellularly, and the exosome analysis using Western blot or ELISA. We expect no difference between the subtypes, but a higher prion expression than in normal meninges, as a past study has shown that meningioma in grade 1 had a higher expression of PRP than normal meninges. However, our subtypes hypothesis is unfounded because studies have never discriminated between the 3 subtypes. However, this analysis may help identify which one has a higher chance of recurrence to allow a better treatment of patients in the future. Second, we will compare the cytokine expression between an M1-like phenotype and an M2-like phenotype via flow cytometry analysis. Atypical meningioma samples will be taken and analyzed to isolate the macrophages and macroglia based on their markers with flow cytometry. There will be in total 4 cultures containing M1 TAMs in an exosome-free media. One will have nothing added as a control. Another one will have bacterial liposaccharide, also known as LPS. A third will have LPS and SPRP. And the last one will have only SPRP. We expect that TAMs exposed to SPRP will have a lower expression of the M1 associated cytokine and a higher one in the M2 associated cytokines. And that is because a recent study found a decrease in the phagocytic activity in macrophages when in the presence of SPRP. Finally, we would compare the PRP expression in the M1 and M2 TAMs derived from atypical meningiomas. We would use the same methods as mentioned in the first step. We expect a significant difference in the PRP expression as M2 macrophages would express more PRP. Prions tend to be more protumoral and help in tumor immune evasion, and thus its higher expression would help the decrease of the phagocytic activity in TAMs and help decreasing the activity of anti-tumoral immune cells. To conclude, our objective is to generalize the effect of prions in meningiomas, as previous research did in gliomas, breast cancer, and colon cancer. We also want to demonstrate that the protein may act as a communication factor between the tumor and immune cells, where it may help the transition from an M1 to an M2-like phenotype of the TAMs. Also, we would like to elucidate the impact of protumoral immune cells on anti-tumoral cells through prions.
Thank you very much for listening. And if you have any questions, we will be glad to answer them. Here's our references.
Thank you very much. I'll open the floor to the judges.
Thank you.
Sushel or Marianne?
Yes, hi.
Off camera, I'm really sorry. I have kids who are using the internet today, and so it's just too much for our poor router. Anyway, I just want to stay connected with you. So yeah, thank you so much for presenting your research idea. I really enjoyed your presentation. I kind of think you have an interesting approach here. I've only ever thought of prion proteins in the context of these extremely transmissible neurodegenerative diseases. And I remember a time we had to dispose of our surgical instrument sets when we did brain biopsies in patients who were found to have these neurodegenerative diseases. So, so this research proposal opened up some really new ideas for me anyway. And it's also an interesting context because, you know, the NF2 gene coding for Merlin as a tumor suppressor is really well implicated in other nervous system tumors like schwannoma and ependymoma. So I kind of thought it was important to explore the relationship between that and tumorigenesis. And it might be applicable to other heritable or sporadic NF-related tumors besides meningioma. So the question that I had for you, just reading through your methodology, so that you mentioned these 3 meningioma subtypes, and these are going to be coming from patient samples, or are there already cell lines, or how are you going to explore that?
We were thinking first to have like the patient samples directly. I'm not sure about the cell lines because, as we said earlier, the studies never kind of like discriminated between the grades. So we would, with consent, obviously, have patient samples that with a pathological like diagnosis right away from a pathologist to really have the different types of brain tumors, the subtypes, so we can analyze them. So we could potentially, with that, as we said earlier, see which tumor might have a higher chance of recurrence just by seeing, for example, the analysis of the PRP expression. So.
I see. And then the other question I had is just about meningiomas and whether they contain microglia. I just wondered if they contain microglia in the way that a brain tumor, primary tumor, like a GBM or something like that might.
Yeah.
Because they're kind of outside of the nervous system, technically. Yeah. So I thought the whole thing was completely logical, but it's just knowing how they arise. I wondered about the immune cells that make up a GBM, that make up a meningioma, rather. Like what are the immune cells in there?
So I can answer that question. Yeah?
Sure, go ahead.
The immune cells in there are every type of immune cells, such as NK cells, CD8 T cells, CD4 T cells, macrophages, microglia. There has been studies that have shown that the percentage of macrophages in meningiomas are about 40% of the tumor weight, which makes it an interesting target because it's the most common immune cells that are in there. So the immune cells that we're going to isolate will be the TAMs in general. That means the macrophages and the microglia, not just the microglia per se. Studies today never kind of like found a way to precisely discriminate between the two subtypes because the markers can be sometimes like wobbly. I know that, for example, they could use TMEM119 to kind of like discriminate between microglia and macrophages, but obviously they express some factors such as CD68 that is common to both types of TAMs. So does that answer your question, or?
Yeah, no, it helps to clarify that. So thank you.
Thank you.
Hi guys, thanks for the excellent presentation.
Thank you very much.
The question I have again relates to clinical application of these things. So if you find the relationship you're looking for, how do you see that as potentially targetable in the future? And in your background reading, you mentioned you found a number of other cancers where the prions have been implicated and are involved. Like, how are they being used in that case? And can you draw parallels?
The prion proteins of other, for example, cancer, as we say that in gliomas, the prion proteins increases with the grade. Breast cancer also have shown, like, for example, whether CD44 receptor is expressed, when they give the drug to the patient that are resistant had a SAR expression even more when they give them the medication compared to patients that are not resistant. So the prion proteins is thought to kind of like help in the multi-drug resistance, as we said earlier. But one of the things that we were thinking about recently, actually, is that we could use silencing RNAs to kind of like go and inhibit the expression of the prion protein. We cannot directly target the gene itself, but we could potentially target the mRNAs. That has to be studied because the prion protein is also an anchor protein in the membrane, and it regulates many pathways. So targeting that protein might not be just enough because some other protein may be overexpressed. So that's why further studies is needed, not only in atypical meningioma, but also in other type of tumors like glioblastomas that also have been shown that other type of schwannomas. So it's really just kind of like opening the floor to kind of like analyze the expression of proteins and its impact in tumor progression. So yeah.
Sure. Important foundational work. That's
Exactly.
Well said. Okay. Thank you very much.
Thank you.
Just one brief point on how you would do this project. It's sort of related to what Dr. Ranger asked you. Do you need fresh tissue for this, or can you use meningioma tissue from a tissue bank? In other words, do one of you need to come to the operating room and collect the meningioma tissue to run the experiments? Or can you go to a tissue bank like the brain tumor tissue bank here in London and get meningioma tissue to run the experiments?
That's a great question. Thank you very much. We were thinking to have like fresh tissues as much as possible, to have things that are very similar to how the microenvironment could look like in the patients. But I think because the studies that have been shown with the gliomas, sometimes they use just cell lines, for example, and compare the different cell line expression between them. And that could be used, just the cell lines could be like a good start, sorry. Then afterwards, we could potentially use fresh patient tissue to have a more clinical application of how we can target, for example, the PRP later in life to kind of see how we can help the patients. So yeah.
Thank you.
Thank you very much.
Thanks, everybody. We'll now move on to the final student team that will be presenting from Memorial University of Newfoundland and Labrador. And you should have the access already to bring up your presentation. Thank you. Ready whenever you are.
Hello everyone, my name is Ali Salman.
My name is Bassel Mohammed.
My name is Abdulaziz Al-Talbini.
My name is Sarah Duraid, and we're all students from the Memorial University of Newfoundland. And today we'll be presenting our proposal of the post-resection radiosurgery and siRNA knockout of EZH2 as a novel approach for the management of atypical meningiomas. Meningiomas are the most common tumor arising from the meninges, which surround the central nervous system. Atypical meningiomas, also known as grade 2 meningiomas, are specifically characterized by abnormal tumor cell appearance, increased aggressiveness, and highly probable reoccurrence, along with higher mortality when compared to grade 1. Meningiomas make up 30% of adult intracranial tumors, and out of that 30%, 17% are atypical grade 2 meningiomas. Even though 80% of meningiomas are seen to be benign, they all share the characteristic of having high relapse rates.
So let's meet our case today. Andrew is a 55-year-old male who has been suffering from symptoms including headaches, forgetfulness, and balance issues. And after running a CT scan and MRI, it was found that there is a large lesion in the vicinity of the atrium of the left ventricle in the brain. He later underwent a craniotomy. However, it wasn't certain if a complete resection was achieved after an analysis conducted on the tumor resected. It was found to be an atypical or grade 2 meningioma. Today, atypical meningioma has a common management plan that consists of 3 methods. Our first-line treatment and first option to be considered is the surgical treatment. However, sometimes the tumor grows in a sensitive region, which makes a complete resection unattainable, in addition to the quicker relapse of the tumor after each surgery. The second option to be considered after surgery is radiotherapy. However, there are 2 limitations to radiotherapy. Although many radiotherapy techniques treat to some extent atypical meningioma tumors, however, due to radiation doses, most of the radiotherapy techniques damage the tissues in the surrounding region of the tumor. And the second limitation is the possibility of a patient developing tolerance to the radiotherapy due to the many radiation doses administered, especially to treat atypical tumors that are known to relapse. The last option to be considered is chemotherapy, and surprisingly, it is found to be inefficient in treating atypical meningioma, where the disadvantages outweigh the advantages, so it is rarely utilized or employed.
So what is the key factor here? Atypical meningiomas are associated with tumor suppressor gene methylation. An enzyme called EZH2 catalyzes the addition of methyl groups to histone H3, leading to a loss of its function. When TSG loses its function, there is a higher chance of atypical meningiomas forming, and hence we would like to inhibit EZH2 and increase TSG expression. This graph depicts the expression of EZH2 in different areas of the body. Notice how the expression is very low. The expression of EZH2 is very low in the brain. When atypical meningiomas form, this level increases. The hypothesis of this proposal is that the siRNA localized knockout of EZH2 reduces TSG's methylation, reactivates tumor suppression machinery, and prevents tumor recurrence. In this proposal, there are 3 objectives that we need to accomplish. Objective 1, we have to examine Andrew to determine if there are any residual tumor tissue to decide if we should pursue proton beam radiosurgery to maximize resection. We'll also need to determine the expression of EZH2 and TSG's pre-treatment using Western blots to have a reference to compare to post-treatment. Objective 2, following complete resection, we will administer gene silencing drugs, aka siRNA knockout drugs. And the final and last objective, objective 3, validating the long-term success of the gene silencing therapy via contrast-enhanced TI-weighted MRI sequencing at 6-month intervals for 60 months, and finally, a Western blot to compare the expression of TSG and EZH2.
It will
In front of us, we have a checklist that summarizes the 3 objectives of our proposal, and our methodology is designed to satisfy this checklist. Our methodology consists of 3 stages, and the first stage is proton beam radiosurgery. Since we are not sure if our patient had a complete resection or not, the latest MR images are supposed to indicate if there is a residual tumor, and in this case, we will be utilizing proton beam radiosurgery to achieve complete resection. Proton beam radiosurgery is painless, non-invasive, and does not damage the surrounding tissues around the tumor, while precisely removing the entire tumor itself. It is very important to ensure that complete resection was achieved beforehand, because our gene silencing therapy doesn't physically remove the tumor, but rather prevents its recurrence. Following proton beam radiosurgery, we are administering our siRNA sequences to knock out EZH2. Administering siRNA alone is inefficient because they can be degraded during delivery and they cannot target our aimed cells specifically. Therefore, we are aiming to use PI-PLGA-based nanoparticle complex to encapsulate and carry our siRNA sequences. This nanoparticle complex can efficiently protect siRNA from degradation and deliver it to the specific cells that we are aiming for, and in this case, it's going to be meningeal cells, in addition to the efficient release of the siRNA sequences to the cytoplasm. Using siRNA designing tools, we have obtained the best 3 siRNA sequences targeting EZH mRNA, and it is also noteworthy to mention that there are several other siRNA-based drugs that are already in the human phase 1 clinical trials to treat different tumors. And finally, we need to assess the success of our treatment using 2 techniques. The first one is T1 weighted MRI to ensure that there is no tumor growth, and the second one is Western blotting to ensure that the tumor suppressor proteins are re-expressed and the expression of EZH2 is reduced. Because we will not have access to the meningeal cells in the patient's brain to conduct Western blot, we will be drawing cerebrospinal fluid that is found to carry our proteins due to its interactions with the primary tumors such as atypical meningioma.
So here's the timeline of our treatment. We will conduct Western blotting pre-treatment so we can compare with the results post-treatment, followed by proton beam radiosurgery if needed before the life-lasting weekly administration of our siRNA-based drug to knock out EZH2. After treatment administration, TI-weighted MRIs will be conducted each 6 months for a 60-month period, in addition to a Western blot analysis to check expression of the EZH2 tumor suppressor protein.
After this timeline, we expect no observable tumor tissue after the weekly administration of our hypothesized siRNA drug targeting EZH2. Here we have some MRI images depicting what we expect a hypothesized patient to look like. Now, there are some differences between this patient and ours, such as the area of the tumor, along with its size, considering Andrew has already undergone a craniotomy, but this depicts our general expected progression of the tumor throughout the treatment. Along with that, we expect the expression of EZH2 to decrease back to the normal ranges. Meanwhile, the expression of tumor suppressor proteins, such as NF2s and DAO1s, is expected to increase after the reactivation of TSGs. Here we have our expected and hypothesized Western blotting for EZH2, NF2, and DAO1. We expect, as seen on the screen, EZH2 to decrease post-treatment, whereas we expect our tumor suppressor proteins, NF2 and DAO1s, to increase throughout the duration of the treatment.
In conclusion, siRNA knockout of EZH2 counteracts its overexpression associated with atypical meningioma, reducing TSG's methylation to the normal ranges. Reducing TSG's methylation reactivates cellular tumor suppression machinery, inhibiting tumor recurrence. Gene silencing using siRNA remains significant around 7 to 10 days. Thus, this is the reason implying the permanent weekly administration of our therapeutical siRNA-based drug. If administration is to be stopped, then AZH2 methylation of TSGs would reoccur. Now, here's our references. We would like to thank Brain Tumor Organization of Canada, Foundation of Canada, and I would like to thank our teammates, and we're happy to answer any questions.
Thank you very much. We'll open the floor to the judges.
Let's see. Hello there, it's Adriana. So yes, thank you, Sarah and Ali and Basal and Abdel Alaziz for presenting your research idea to us today. I really enjoyed your presentation. To me, your concept sounds quite logical, but I'm just curious to know why you selected proton beam radiosurgery for your study proposal, since this type of radiation treatment isn't available in Canada yet, at least not to my knowledge. Certainly it has advantages of sparing unaffected tissue and the side effects of radiation toxicity, but right now it's only approved for some pediatric cancers and some adult cancers with curative intention. So I'm not sure how a trial like this would work, or if you would substitute another radiation modality.
So I'll be answering this question, and just to summarize the question. So your question is basically, why are we using proton beam radiosurgery rather than other radiotherapies that are well used and well known more than the proton beam radiosurgery?
Well, yes, but moreover, I think that the justification is very clear. And certainly I take care of children who are often sent to the American centers for proton beam therapy for their brain tumors, but we don't have this yet in Canada. I understand that Toronto may get a unit, but if we're going to do this sort of research in Canada, we would have to be sending patients to the States for their clinical trial. I don't think Health Canada would support that. So maybe another modality of radiation therapy could be considered. It just came immediately to mind because of the population I treat anyway.
So the question for this, as we said, like proton beam radiosurgery has so many advantages compared to the other radiotherapy techniques. And yes, that's actually a fact that proton beam radiosurgery has a limitation, and this disadvantage is the fact that it is inaccessible. However, in our proposal, proton beam radiosurgery would only be used for our case study in case there was a residual tumor, like if there wasn't complete resection achieved at the first place. But if there was complete resection, we wouldn't even go to this step. So this is just a precautious step in case there are any residual tumor tissues left. In case there are any residual tumor tissues left, there are other radiotherapy techniques that can be used. There are so many articles like listing all the radiotherapy techniques. And I believe one of them was fractionated radiotherapy because it causes, I believe, the less damage to the surrounding tissues. However, if it takes a patient to go to the United States to get a radio beam radiosurgery, I know it's something inaccessible. However, yeah, proposal-wise, I believe it was our main focus on proton beam radiosurgery because of the advantages it gives.
Okay, thank you very much.
Thank you.
As the radiation oncologist in the crowd, I had similar questions along those that Adriana asked. Your use of radiosurgery implies that it would be a single treatment. This is not, to my knowledge, common in proton beam therapy, and certainly single treatment adjuvant therapy for photon radiation, which is the more common approach in Canada, is also not common in this setting. We would tend to use, as you said, a conventionally fractionated course. Why did you choose the radiosurgery method rather than sort of a standard? And I guess this is getting to my point of your study is changing, or you're investigating sort of two things: the use of proton radiosurgery, which is non-standard, and then also this new compound. And so there's a couple of things changing in your protocol, which can potentially mask results. How do you respond to that?
So to summarize, your question is, why are we using like one session of radiosurgery instead of like the standard multiple sessions?
Yes.
Okay, so for in our like proton beam radiosurgery is usually used like to resect the tumors. However, in like using the radiosurgery, definitely. However, in this case, the tumors are already there. But in our case study, Andrew has already like done craniotomy, so the tumor is already resected and only like residues are left. So we believe that one session of proton beam radiosurgery should be enough to remove any residual tumors in case there were any. I'm not sure if we're understanding the full image, but like based on the readings that we've done, one session of proton beam radiosurgery should be enough to ensure complete like removal of the tumor, just in case if there are any residual tumors left.
Thank you. Joe, do you have a quick question? We are up for time, but.
Sure. I mean, I think, just to clarify, I think what Adam was saying, and I kind of agree, it seems that you've kind of pulled this proton beam out of a hat a bit. It sounds neat, and but it's not really standard care. Even in the US, they have a number of machines, but certainly not as many as conventional radiation machines. And I think sometimes, this idea of radiosurgery versus radiotherapy gets mixed up. So I agree with Adam. I think if you're going to look at EZH2 or EZH2, I guess as we would call it in Canada, as well as using a proton device rather than a conventional device, it does make the data hard to analyze. But anyway, it's still an interesting project. Thank you.
Thank you very much. And thank you so much for all of the teams for taking the time to present. I think I've got a bit of an echo there.
Thank you.
Thank you.
We will now pass it along to Dinah, who is a studentship recipient for our 2021-2022 studentships, and joins us from Dr. John Trant's lab at the University of Windsor. Dinah, as you're getting set up, I'm going to get the judges sent off into their own room so that they can deliberate. So just give me a moment while you're pulling up your slides while I do that.
Sure. Do I just start sharing my screen?
Yeah, you can do that. You should have access.
Sounds good. Thank you.
You're welcome. So judges, I'm going to whisk you away, and if you can please join us within the next 15 minutes, that would be greatly appreciated.
I'm ready to start whenever.
Okay, perfect. I'm just making sure that the judges have headed off, which it looks like they are. Is that true, judges? Can anyone hear me from the judging panel? Excellent. So whenever you're all set, Dinah.
Okay, perfect. I will start presenting them. So hi guys, my name is Dina, or my real name is Dilnar. I'm from, I guess, University of Windsor, Dr. Trant's lab. I graduated recently, and I am honored to present here as the recipient for the 2021-2022 Brain Tumor Foundation of Canada studentship. And my project that I have had the studentship for is known as, or is called Self-Immolative Nanocapsules, a new form of drug delivery for glioblastoma multiforme. So a little bit of introduction about myself. Also, I need to find the laser pointer. Okay, there we go. So I recently graduated from my Bachelor of Chemistry degree at the University of Windsor, and I love organic chemistry as well as photosynthesis, also catalysis. Chemistry in general is just really intriguing to me. I was also a Uyghur international student from Xinjiang, but I am not a student anymore. However, I was in Dr. Trant's lab at U Windsor throughout my 4 years of undergrad, and now I'm continuing to work there for him as a research assistant. And yeah, I got the studentship for the past 2 summers, and these are some pictures of my chemistry and myself in lab. So let's start by asking the question why, and what is glioblastoma multiforme? This is the most common type of malignant brain tumor. It also has a 5-year survival rate of under 7% and 10-year survival rate of under 1%. It's almost always lethal, and surgery can remove it, but there are problems with surgical removal, including that, as we all know, brain is a sensitive and complicated part of human body. Taking any like healthy tissues from brain might cause permanent damage. So it's always really finicky to like do surgery on brain tumor, at least to remove it completely. It can respond to anti-cancer medication. However, there are problems associated with that. And some of them include the blood-brain barrier, which, as you may know, it's a highly selective semi-permeable membrane. It can prevent harmful substances from entering the brain, but can also prevent helpful drugs from entering the brain. And then there's also the issue with polymorphism, meaning there are many mutations that it's possible that exist for GBM, and this causes it to be hard to target. And these are in addition to standard treatment problems such as premature drug release, meaning that the drug is released in human body before it reaches the target site. So the drugs that was released will kill some healthy cells along the way, which causes cytotoxicity, which is, you know, side effects that we don't want. So there are other systems that were developed for drug delivery purposes, including, for example, liposomes. But these nanocarrier drug delivery systems have the issue of, you know, they might break down in human body or even before human body. So the liposomes are entropically favored to rearrange into smaller liposomes. So if you put drugs in them, it's easy that they just, you know, break down and then the drugs will be lost. So even if they can cross the blood-brain barrier, they might also cause premature drug release, leading to cytotoxicity issues. So for that, our solution that my lab proposed is designing something known as the self-immolative nanocapsule, or SYNC. Now, this is a drug-bearing nanocapsule, so imagine like a little ball, and then in the core, we encapsulate the drugs. It will provide, in theory, stability during transport inside and outside of human body, so it's not supposed to decompose and, you know, release the drug prematurely before reaching the target tumor site. And it's composed of hydrophilic and hydrophobic components. So I made this little diagram myself, but it's actually not the most accurate representation. It should only, you know, have one side of hydrophilic, not both sides. So ignore this, the left part. And hydrophilic meaning, you know, likes water. Hydrophobic is like doesn't like water, so like, you know, dissolving in oil, that kind of stuff. And then we also proposed that we could attach some surfeit peptide sequences to the hydrophilic component in order to enhance the ability of the SYNC to transport across the membrane or some protein channels, and also be used for cellular recognition. The other thing that we try to do is have this, I guess, yellow lines in my drawing. This is known as cross-linking. So imagine, you know, a monomer being a pearl or a bead, and then a polymer is like you have a string of beads. But if you only have, you know, 10 or 20 or 50 of them, they are not stable. But if you cross-link them, meaning if you attach the strings to each other, then it's like literally unbreakable. So we propose we can also cross-link the polymer backbone so that they are stronger than just a micelle. But then the issue is, well, if it's so stable, how are we going to release the drug once it reaches target site? Because you don't want something that doesn't degrade eventually. You want the drug to be released. And for that, the solution is using self-immolative polymer, or SIPs, as the backbone of the nanocapsule. And this, we would be able to achieve degrade on command at tumor sites. So self-immolative polymers is something that's, you know, that has been relatively new in the polymer science field. And my professor, Dr. Trent, has done his postdoctoral fellowship at Western University studying self-immolative polymers. So this idea was really cool. Like, you know, I was kind of like blown away when I had the proposal idea from him. And then also the other thing that we are really trying to focus on is biocompatibility. So the self-immolative polymer backbone, all the monomers are made of amino acids. So once they break down, they will break down into amino acids, which are natural to human body and will not cause any other like toxic effects to human body. So like I mentioned, this is the polymer backbone, and I want to just go into a little bit detail about the self-immolative polymer component of the nanocapsule. So cross-linking makes the vesicle impossible to break, and this is undesired because you do want it to break at some point and release the drug. So the solution is the backbone being degradable. And these are unstable polymer chains that is being held stable by something known as end cap. So without the end cap, it would just like self-destruct. It will degrade in a chain reaction fashion and break down to monomers. The end caps is the only thing that is holding it stable. And then these end caps are stimuli sensitive. So if you apply a certain type of stimuli, the end cap can be cleaved and the polymer can be degraded on command on site. This is the whole idea behind this project, basically. And then, like I said, amino acid-based SIPs should have biocompatibility, which we do believe that it has. So my previous work this is a 2-summer studentship project. In last summer, I was working with this PhD candidate called Mana on this project. She was, leading me through steps and stuff. We were able to synthesize the sorry, I see a chat. I just want to make sure that people can hear me. Oh, never mind. So yeah, last summer we were able to achieve the synthesis of polymers using 3 different diamino acids as the monomeric building blocks. And then each one of these synthesis is 5 steps. So it was like kind of a lot to make backbones from 3 different amino acids, but then in theory they should all work. So we wanted to try all 3. And then after we built the polymer, Mana performed studies called like GPC, or gel permeation chromatography, which is supposed to tell you the degree of polymerization. So a simple flow chart on the left-hand side will explain how this works. You have a mobile phase, which is usually the organic solvent that will be injected into solvent delivery pump, and then which then will be transferred to the sample injector, which includes the sample also via injection. And then the mobile phase solvent dissolving the sample or carrying the sample will carry it through GPC column, which is also known as the stationary phase. And then after the column, there's a detector that will detect the molar mass or molecular weight of the compound, of the polymers. And so by dividing the mass of the monomer in theory over the overall mass of the polymer, you can get degree of polymerization. And so the degree that we obtained from our 3 polymer backbones were anywhere between 70 to like 300-ish, I believe. The exact numbers are like, I'm not clear with that because she did all the GPC studies. This is kind of complicated and I wasn't like, trained to date to do this yet. She also performed depolymerization later on by applying heat because our end caps last summer was heat sensitive. And then she was able to show that the polymer backbone was able to degrade on command when you apply heat to cleave off the end cap. So the conclusion we had was thermal sensitive end cap was able to function properly. And then this backbone did do what it's supposed to do. So in theory, this works. And so what happened this year was Mana, she recently had a baby, which is really awesome. But she kind of left. I'm not sure if she's coming back to finish her PhD, like defend or not. We're unsure about that. Hopefully one day she comes back. But this summer I was kind of, it was a little tough because I didn't have enough, like, as much guidance as last year because she was gone and I was the only person on the project. I mean, I had my own little team of students that I trained, but this is the background of it. So I'm trying to redo the whole thing. And the other thing that happened was the polymers that we made last summer decomposed over the past year, which is also not uncommon in organic chemistry because things are not that stable sometimes. And especially, things that are like this that are meant to degrade and they're not very stable inherently. So it has degraded. So I had to remake the whole thing. It's a 5-step synthesis. We use various organic, like synthetic chemistry techniques and also reagents and protecting group strategies. But instead of using 3 amino acids, I kind of just focus on one of them that we think would work the best, meaning it will degrade the fastest, like once it reaches the tumor site. And I just want to mention that the degradation mechanism is via cyclization. Also, this is a little, I guess, diagram showing how the monomer looks like. I cannot disclose the structure, unfortunately. I would love to because I'm a synthetic organic chemist and all I do is, I show people my synthesis procedure, my structures, my reaction schemes, how this works. But unfortunately, due to I think this work might get published or apply for patent in the future. So all the IP issues, my professor said that, I shouldn't disclose the structure. So we're just representing things with shapes and blocks and colors. So this is not easy for me, bear with me. So the yellow part or orange part and this thing is the amino acid, which is the monomer. And this is actually the amino acid monomer at the end because we have an end cap and not all of them have an end cap. Only the terminal ones should have the end cap. The amino acid is also linked to two other things. The first one known as self-immolative linker, and then the second one is variable functional domain. So self-immolative linker is the thing that's supposed to cyclize and decompose once the end cap is removed. And then the variable functional domain can literally be anything. In our case, it's a protecting group because, as you know, amino acids, and we have diamino acids. So sometimes you want certain part of the molecule to react, but the other part to stay inert to specific chemical environments. So you have to protect them selectively and then only allow the one part to be exposed to the UV agents. So they do what they're supposed to do. And then in our case, the functional domain is protecting groups. The thing attached to the self-immolative linker is known as activating group. And this is the thing, or in our case, actually leaving group, if you remember organic chemistry 1. This is the thing that's supposed to cause monomers to build into polymers, and they will leave after the polymerization because that's what they do. They're leaving groups. But yeah, this is the structure. And then I want to talk more about the end cap because this is something really neat. This is what gives you the idea of precision in drug release. So end caps, like I mentioned, are only responsive to specific stimuli. And this is great because human body is very sophisticated and complicated. You don't want them to to drug release prematurely because, like I said, that leads into toxicity effects. And if they're only responsive to one thing, that'll be great. So I'll explain more in my future slides about what we chose to be our stimuli to cleave off the end cap. And yeah, so cleaving off the end cap will cause decomposition of the polymer backbone once the nanocapsule is at tumor site and prevents premature drug release. That's the idea. So we had two approaches for the end cap moiety. But before that, I guess I will talk about in general some of the end caps that exist in the literature that are most commonly used. They include pH, so acidity/basicity, light, temperature, or enzyme, etc. And our focus is thermal sensitive end cap and light sensitive end cap, as you can see from the little picture popping up. We didn't decide to go with pH because, well, human bodies have acidic parts like stomach and basic parts like intestine, and maybe I think lungs are also slightly basic because of the sodium bicarb concentration. So if you do, pH sensitive end cap, it's too risky. It might release anywhere else. You don't want that. So, sorry, a breakout room thing just popped up. So yeah, it is ideal to do the heat just to see that it's doable. And then the light one, there are 3 major advantages of using light sensitive end cap. So the first one, which is really cool to me, although I don't really, I'm not a biologist, I'm not going to pretend that I understand a lot of biology. I try my best, but this is outside of my expertise. The first one is known as xenograft study of grafting human brain tumor cells into zebrafish. And this, we are collaborating with the Porter Lab here at Windsor, which are excellent researchers doing cancer research. So they have developed a whole set of zebrafish model that they study tumors and cancer with. So some species of zebrafish, their skin is transparent. And by having a light sensitive end cap, you can, in theory, just shine a light into, I don't know, the fish tank or something, and then you can detect the decomposition of the nanocapsule and release of drug, which is really cool. So this is methodology development as well as proof of concept. We can prove that, oh, this works because, you can directly detect from the living model that it works. The other really cool thing is known as, or called personalized medicine. Like I mentioned, GBM has a lot of mutations, and you might have different patients with different mutations of glioblastoma multiforme. So it is also in theory possible that grab some, brain tumor tissue from the patient, and then you apply them to a cohort of zebrafish and have nanocapsules carrying different trial medication, chemotherapeutic drugs in capsules, and apply them to specific zebrafish and see which fish or which drug, I guess, responds to that patient's specific type of brain tumor. And this is the idea of personalized medicine. You can, in the sense, achieve maximum proficiency by finding out, oh, this drug really works well for this type of GBM or this patient in particular. And then the third thing, which is the most obvious applications of light sensitive end cap, is intraoperational work. So during a procedure or a surgery, can just put the nanocapsule there via injection or something, and then like shine a light on the tissue, and it just like decomposes there and release the drug on tumor. So this is what my professor has told me, and I think it's really cool. So I just thought to throw the application link there for you guys. And the last thing, kind of the last thing I want to mention is how do you make the hydrophilic component attached to the hydrophobic part? So there is a chemistry, I guess there's a field of chemistry known as click chemistry or click reactions, and these are high yielding reactions with no need of purification. It's simple to perform, it works, it just clicks like that, which is where the name came from. And the most famous class of click reactions is known as copper-mediated aza-alkyne cycloaddition or cyclization. And these type of reactions happen between alkynes, which is CC triple bonds, and azide functional groups via the mediation of a copper-1 catalyst. And so this we propose to attach the hydrophilic component of the polymer to an alkyne and then make the light-sensitive end cap with azide functional group attached to it. And then you can just click them together and then make the hydrophilic component be attached to the hydrophobic part of the whole polymer. And now this is known as amphiphilic polymer, which is good because then it will self-assemble in solution to give you micelle, which is, like I said, the next step. So how to make it, or I guess the future steps, where I am at right now, the overview of the project. Although my studentship, I guess, term is over, I am still pursuing this project. As far as I know, until, you know, my last day with the lab, I'll still be working on this project because it's so important and Dr. Trent wants to, we all want to see it succeed. This is great. It's just, it's hard. 90% of chemistry, organic synthetic chemistry is failure. 10% you might be lucky and it might succeed. But this is just, if it's successful, it will be the first like of its kind of drug delivery mechanism in the world. So we are definitely like, you know, we have high hopes for this. It's just, it's not very easy. It's not trivial. The synthesis of CYP monomer has been successfully achieved. And then the backbone, we made it last summer. So this summer, I'm trying to replicate that chemistry. Also, we're trying to synthesize the light sensitive end cap, which is completely brand new. Last year, we didn't have this idea. So me and my team were working on this, but this is also a little finicky because the last few steps of the synthesis have to be done completely in the dark because it's so light sensitive. So we have to, you know, turn off the lights in lab and wear night vision goggles or like wrap at everything in aluminum foil, and so they don't get exposed to light. So it's taking time, but we believe it is very like achievable. After the end cap has been attached to the backbone, we will perform cross-linking to the, like between the polymer backbone, so between the string of beads, and then functionalize with click chemistry to attach the hydrophilic component. And then self-assembly in solution, which I think will not be hard because, but famous last words, I think it will not be hard. We have excellent, you know, polymer research groups here at UWindsor who are experts in self-assembly chemistry. And then after that, we will do in vitro and in vivo testing with the Porter lab. So either with or without the drug, and to see, you know, if it degrades, if the drug releases, and all that like biology stuff in the future down the road. So I just want to say thank you for the Brain Tumor Foundation of Canada for supporting me with the studentship. And I honestly am deeply like honored to be awarded this. It's a very, it kind of changed a lot of things for me. Because as an international student, first of all, I wasn't eligible for a lot of things like NSERC summer scholarships. I cannot apply. But I was very happy that I got this so I can do what I really want to do for 2 summers. And then I also just want to, I guess, let you guys know that, you know, go out there, email professors, meet with them, talk to them, and try to join research labs. I didn't think these things would be possible 4 years ago or even 2 years ago. And here I am giving an update on my research to all of you guys. And I've heard some of the competition that happened earlier. It's like really impressive. And I really hope, you know, everything seems impossible until you've done it. So get out there, make connections, talk to people, start research, and maybe someday you guys will be able to solve the cancer problem. And I have hopes for that. So thank you for listening, and I will be able to take up any questions now.
Thank you very much. That was a great presentation, and we're excited to hear all the great outcomes coming down the pipe from you guys. I appreciate that we are a few minutes over time, but we can open it up just for a couple minutes of questions if anyone has any questions. I would just ask that you unmute yourself and you can ask those. Okay. Well, it looks like you were so thorough. Nobody has any questions. If people have anything, then please do feel free to reach out to myself, and I'm happy to ensure that those questions are answered. Thank you so much for taking the time being with us today to present your work, and we look forward to, as I said, your report on this previous summer's work, so that we can share that accordingly on our website. So thank you very much. I'm now going to pass the baton over to Adam, who has got the results from the student competition and will share those with us. So Adam, I'll pass the baton over.
Thanks, Sue. I just wanted to start by saying thank you to everyone for coming today. This is an excellent turnout. I wanted to thank the Brain Tumor Foundation for sponsoring and organizing this. A big thank you to Sue, who puts in a lot of work on this, and to my fellow judges. We had an incredibly difficult time this year picking our winners. It's tricky every year. I think I say it's the trickiest every year, but I truly believe that this year was the most difficult of all. As Adriana alluded to earlier, there were many, and Sue, I suppose, as well, but there were many applications that had been screened out, letters of intent that we had reviewed and didn't make this step. So first of all, congratulations to all the teams for making this far and going through the process. That's a big step. The quality and detail of the research that was done and presented is always far beyond what we expect. I'm very impressed, and I find I learn a lot doing this every year. So thank you all for that. I think this year in particular, the quality of the presentations, both the slides and the way they were delivered, were particularly strong as well. So people are perhaps getting used to Zoom and mastering that. So well done to all of you. I think it's important to note, whether you win or not today, the process of doing the work, preparing the proposal, the background research, putting yourselves out there and preparing your presentations is an important first step as you sort of discover what your future research will look like. And I encourage you all to continue to keep at it. This is the energy and ideas you're bringing are invaluable and are what are going to make a difference in the future. So I commend you all for that. The results today, in third place, we have the team from McGill and their Quality of Life app. If you guys could just turn on your cameras and wave so we know you've heard. Okay, congratulations, guys. For the first time ever, the judges were unable to come to a decision about the top team. And so there will be no second place winner, just 2 first place teams that will share the prize today. And those are the teams from Western and Queens. Congratulations, guys. Exceptional work, and to yeah, it was very well done.
Excellent. Thank you so much, and congratulations to the winning teams. I said to Adam that the judges always have a tough decision and always make things unique in the end. So thank you so much for taking the time. I know that it's not an easy decision for all of you to come up with the top candidates when they're all so great. So thank you everybody for taking the time to spend with us today, for your time in creating your letters of intent, for those who move forward to today's presentations and presented today. Congratulations to the winning teams. I will be in touch with you probably early next week to speak about moving forward. Thank you to all of the participants who joined and supported the teams today and being here to listen to the presentations. A special thank you to the Del Maestro family who make this the awards possible, and the monetary the gifts that the top teams receive. I think that regardless of everyone's status today, everyone most certainly has received bragging rights to the work that went into the presentations, and your institutions should be pleased to have you being the leaders today and representing your educational institutions. So great to see the brilliance always around this table, and to know that we have such a great future in store for brain tumor research. And I think that we're in great hands with all of you as we move forward. Again, teams, I'll be in touch, and thank you all for making today such a great event. I apologize that we went about 10 minutes over, but I wish you all a wonderful remainder of your Saturday. Thank you again, and congratulations.
And thank you, Sue, for organizing it. I know it's a lot of work, and keeping Adam and I and Adriana on track can be a challenge, and keeping all the students organized. So thanks again for doing this.
Thank you very much. It's my pleasure. Thanks, everybody. Have a great Saturday.
Take care.
Bye-bye.